All right, excellent.
Excellent. Okay. Sorry about that. Thanks for the introduction. I'm sorry for wasting a minute or two there. Also, thanks to the Neuromatch organizers for putting on this greatly needed meeting. So yeah, I'm a computational neuroscientist. Much of my work is focused on theories of the neural computations underlying spatial cognition, including interactions between place cells and grid cells. But in this talk, I'd like to describe some data and some modeling that potentially supports a theoretical role for septal neurons in maintaining the allocentric reference frame of the spatial cognitive system, at least in rats. Particularly, I'm going to focus on the question of path integration and how you might be able to maintain an allocentric reference frame during path integration, which basically comes down to the problem of how do brains reset the accumulation of encoding errors. So path integration, as has been described, is the critical component of navigation that contributes spatial information by integrating self-motion signals. Resetting is necessary because integrating self-motion accumulates errors in position estimates over time. And that's kind of illustrated by this green trajectory here shown for this journey. And so there's a lot of different ways of doing that. There's been two main kind of camps, theoretically speaking, for how to model path integration and how it might interact with landmarks and cues in the environment. One of my much older papers kind of explored the oscillatory interference mechanism, which is where if you have theta rhythmic oscillators that are also modulated by direction and speed, then you can combine them in different ways to form grid cells, for instance. Those were models from John O'Keefe and others, and Neil Burgess. But I showed that you could generalize this or genericize this into a place field, more of a generic spatial mapping model, where if you randomly, if you have random preferred directions with these VCOs, these velocity controlled oscillators, then you can build place fields that are stable and based purely on path integration. And this was work with Jim Knierim's lab of modeling his data on circular tracks. And so this is just kind of showing how the amplitude of synchrony can map into space through path integration with oscillators. But one of the things that I was really concerned about in this paper was how does a sensory cue actually feed back into the phase code in order to stabilize a phase code from drifting away. And so in the bottom panels here, you can see that if we posit some amount of phase error and we want that to be corrected at some point in time, that represents the interaction with an external cue, then I simply posited a very kind of abstract feedback process represented in this diagram on the bottom right, which is what I needed to be able to study this in terms of remapping and partial remapping. But this was definitely a black box. So this kind of really raised the big question, like, is there an actual neurobiological basis for having this kind of spatial feedback, particularly in the phase domain? So the question is there a phase code, an oscillatory code outside of, potentially outside of hippocampus that could serve a resetting function for path integration? And so this data is a result of a collaboration with Tad Blair's lab at UCLA, where he performed these very long duration recordings in an 80 centimeter cylindrical arena, basically standard random foraging tasks, while recording a reference hippocampal LFP signal. He recorded essentially from all of the subcortical brain areas highlighted here that are in one way or another interconnected with the hippocampal and entorhinal formation. And so he recorded from all of these areas, and I basically took this into a data analysis paradigm to look at the amount of spatial information carried in phase, in the theta phase, as well as correlating that to velocity and position and other characteristics. And I just to kind of give the hint of where things are going, I found this kind of spatial phase code, which I think might serve this theoretical role that I described, only in one place, and that was in lateral septum. And lateral septum happens to be the primary subcortical output target of the hippocampus. So it's possibly the entrance point to an interesting feedback loop if we consider all the interconnectivity within these networks. And so I'm going to take you through what some of this lateral septal data looks like. And just as a brief overview, if you record what a theta signal is, probably this crowd doesn't need that, but you can record the LFP, you can do a bandpass filter, find where the peaks are, and then we can take the spike timing from relative to peaks to peak within each theta cycle, and we map that to the phase domain that goes from 0 to 2 pi. And just to be, so that explains the y-axis of a lot of the plots that I will show. And so here's just a standard spike trajectory plot of one of these cells. And you can clearly see there's, this is a very long, I think a 2-hour recording. So you've got the gray trajectory there with the random foraging and the red spikes. And you can see clear spatial modulation. So if we look at the top map here, you can see that the firing rate, clearly illustrated. There's a broad place-like field and on the west side of the arena, which, that's great. We got spatial modulation in lateral septum. That's been shown sparsely in the literature, but that has been shown before. What hasn't really been shown is that relationship to the ongoing theta oscillation in the hippocampal system. So if you look at the bottom map here, this is a phase map of the average phase at every location. And you can see that there is a correspondence between the pattern of modulation in the rate map on the top and the phase map on the bottom. And so this is interesting because this is over a very long period of time, a very long recording, and there appears to be a very strong relationship between rate and phase. So that's kind of the basis of the idea going forward. And I quantified that by looking at the phase rate correlation, the circular linear correlation for basically the pixels in these maps. And you can see what that looks like. So you get kind of the expected negative phase rate relationship. And I termed these cells, I called them phaser cells. So these are lateral septal phaser cells, for want of a better word. We'll see if it catches on. But basically, we're going to analyze that correlation, but the correlation kind of immediately brings to mind that you can explain this with a fairly simple phase coding mechanism. So if you posit that there's some cell that it receives an inhibitory theta rhythmic input, such as the magenta sinusoid that you see here, and then you also posit that, well, maybe it also receives a slowly changing or slowly ramping depolarizing input, like the green triangle wave here. And that's all you need to get kind of the phase coding relationship that we just saw, where as the input increases, you start to get activity in the cell, and then you get more and more activity within each theta cycle as the input goes up. But then the activity within each theta cycle also occurs at an earlier time, or it initiates at an earlier time. So you have this joint modulation of phase and rate. And critically, as once an input slowly ramps up, it can slowly ramp back down, and symmetrically you see the same exact thing. The phase will deflect all the way back up to the baseline phase once that input has gone away. So there's no kind of hysteresis or learning going on here. So this is a symmetric bidirectional phase coding mechanism, and something like this has been posited for phase for place cells in hippocampus before learning or any kind of network effects have come into play. But the idea is that this gives you a scalar code. So this is basically the co-modulation of phase and rate means that the phase is basically just a conversion of rate. It's analogous to taking the spatial information in a rate code and then putting it into the phase domain. And so we can think about functionally, why would you want to do that? And it is in particular a high contrast with what you see in typical place cells in the hippocampus. So on the right, I've taken a figure from Souza and Tor to the 2017 paper where they analyzed a large place cell data set and showed this kind of canonical unidirectional asymmetric relationship between phase and rate. So as the animal goes through a field, the phase will continually go down and it does not return. However, if you had a phaser cell field, as the rate goes up, the phase would advance, and then as the rate goes down, as the animal leaves the field, the phase would delay back up to the previous level. And so looking for this type of phase code, I set up a number of different criteria, well, 4 criteria, 3 of the important ones. So looking at the spatial phase information, looking at the total phase shift, like how much does it change within that correlation, and then the strength of that phase rate correlation, essentially. And then using those criteria, I was able to filter TAD's entire data set, the subcortical data set, all those single unit recordings into cells that meet these phaser cell criteria and those that don't. And particularly, it's important to look at whether this is actually a stable code or not. So just looking within session, I compared up to the first hour of a session, the early part, up to the last hour, the late part. And so if we look at on the left, both a spatial correlation, and on the right, a big change in that total phase shift. This kind of illustrates that you do maintain spatial correlations across these long duration recordings, and the phase shift that comprises or that constitutes that phaser cell code does not significantly change. Most cells remain within about pi/4 or about 45 degrees across these multi-hour recordings. And then you also want that to exist across days, and that is basically what we found with the curves looking very similar. And between days with identified units, we can kind of track what each individual cell is looking like across days. And the vast majority of them do not have significant changes or flips in the direction in their phase shifts for this phase code. A couple of them do, but most of them are pretty stable, which is pretty good. That's what you want to see. But then the last thing besides stability is that you want to make sure these spatial responses really are spatial and not just a confound of spatial correlations of other aspects of the trajectory. So to kind of deconfound that, I trained a GLM, a generalized linear model, with both spatial predictors and trajectory-based predictors. And so these variables called LMQ, or just linear and quadratic. So this is up to second order spatial variation. And then the trajectory predictors are wall distance, speed, and direction, basically. So this top grid, this top grid is showing you that the responses are utterly dominated by the spatial factors and almost not at all the trajectory-based factors and other confounds. And even if we look at the maximum possible contribution that each of these predictors made, which is the bottom plot here, you still see that dominance of the spatial relationships, though that does kind of also reveal that there is this hint of a trade-off between how spatial the cells are and then how tuned to speed they are. If you look at the sorting along the fourth column with S here. I'm not sure if people can see my mouse, actually. I'm waving my mouse around. But, so once that's kind of established that these are spatial cells and they are pretty stable, we have those criteria. We can kind of see where these cells fall. So on this plot, I'm showing all cells that had significant spatial phase information. And so that spatial phase information is on the x-axis, and then the total phase shift, that phase modulation, is on the y-axis. And you can see we have these cells with a negative phase rate correlation here in this bottom part of the plot. And these circles are, the size of the circle correlates to how strong the correlation is. So you have these like nice strong phase rate correlations showing these strong negative phase modulations, giving us lots of information about space and phase, which is great. But then the surprising thing is that if we look at the top, we also saw cells. So the cells up here with positive phase shifts. This was surprising because of that simple mechanism that I implied earlier, that I described earlier. You wouldn't expect higher firing rate to correspond to later firing in that intuitive model. So there's probably something else going on here, and this potentially has interesting implications. So just to show some examples, these are 5 different examples of those negative phaser cells from different animals. The top row shows you the rate maps, the middle row shows those phase maps, and then the bottom shows the phase rate correlations, just like the example cell that I showed earlier. And you can see there's like wall responses, there's place-like responses, place boundary conjunctive responses, and then kind of like broader, but still spatial responses. So there's a strong diversity here.
Joseph, you have 1 minute left.
Darn, okay. And so here's an example of the positive phaser cells, which are not as strongly spatial. So if you look at the phase rate, the trajectories of these cells, you can see across phase, they interleave very nicely. You can see in this plot here. And then if you look at the typical firing phase of these populations, you can see just how interleaved they are. So at any moment in time, you've got information coming in the phase domain about space from one population or the other. And then so very quickly, the modeling of this is that I had a dynamical circuit model with a very simple structure here using feed-forward suppression of the negatives, of the positive cells by the negative cells. And you can get this complementary relationship pretty much exactly, which is very nice. And then I use that GLM as a generative model to generate spatial tuning curves for both 1,000 negative phasors and 1,000 positive phasors. And I use those as an input layer into a set of downstream theta bursting target neurons. And so these are just random target bursting these are random theta bursting neurons that are not path integrating or spatial. The question is, can we make them reset to a path integration signal? And so with different types of supervised phase codes, which I won't get into detail because I'm running out of time, we can actually see how well these codes were learned by these downstream target cells based on a very simple competitive learning mechanism. And so once you have that, we have a very small number of these cells. You can actually do population decoding of just the phase. And you can see that, well, this top structure didn't work very well, but the bottom one did. And that actually is going to lead to a very rapid phase resetting mechanism, basically a sub-second reset mechanism for path integration. So that's basically the idea that we've got the fairly simple network structures and circuits, and using this kind of like location-based synchrony idea, there might be a subcortical pathway for phase code feedback in the spatial system that might support path integration and other elements of navigation. And I will end there.
Perfect. Okay. So we have to move swiftly on to make sure that Balázs has enough time for his talk. You do have a few questions in the Q&A from Eleanor, so I would encourage you to go check a look at that. And thank you again for this great talk. I'm going to move swiftly on.